Um, to answer Sudarshan, if the webinar is going to be audio based, no, the webinar is going to be both visual and audio based. Iftikhar, we will be getting to the course fees at the end, so we will be answering all of your questions. To answer Sudarshan, if you have any other questions, please let us know. Yes, Sudarshan, we will be sharing our presentation just in a minute. And we also have a YouTube link for anyone else who wants to watch it on YouTube. So we will be streaming live. Yes, Iftika, you can join it now. Yeah, we will, Hanif, we will be answering all of these questions. So please bear with us for the next 10 more minutes as we start the program. And we will be answering all of your questions and also have a discussion on how we can use bioinformatics for biology and discussion about infectious diseases. Okay, yes, I have a lot of questions in the chat. Okay, Abhimanyu, Sudarshan, yes. So we will answer everyone's question and you can definitely ask us any question. Please put them down in the chat box because we will have dedicated time slots where we will be answering each of your questions. So please keep posting all the questions that you have in the chat box. If you want us to reach out to you, you can leave your emails as well. And if you want to reach out to us, here is our email ID, marketing@pine.bio, where you can reach out to me for any kind of questions. And we will be getting back to each of you and also have a discussion today with all of you regarding this subject. I hope everyone can see my screen clearly and you can hear me also clearly. If you have any kind of audio problems, please let me know right now so that we can help you out. And you can always use the Zoom audio option in case you're not able to hear me. Thank you so much, Kirivasan, for confirming that. We just want to make sure that everyone can hear us and there are no possible technical disturbances in between. So we have 6 more minutes to go. Thank you, sir. Thank you so much. It would be great if everyone can keep their microphones muted when a speaker is speaking during this session. And we will take all of your questions in the chat box and maybe have a dedicated time where you can actually unmute yourself and ask questions. You can just let us know that you have a question. So we will be allowing that time for you. Yes, so we kind of keep it muted so that there is no kind of technical problems during the session, because oftentimes with the video running, many people feel it's disturbing them, or sometimes even the sound can be very disturbing. But we will try to make the webinar as interactive as we can and also hear from your end. Thank you so much for understanding. Yeah, and thank you, Mohammed. We hope that you enjoyed today's session. Yes. Hi, Kaiser. So we are going to be beginning in just a few more minutes, and you can use the chat to ask any questions you have for our speakers who will be interacting with you during the session. And also, if you have any questions other than what we talk about in the session, we have an email ID you can reach out to. And you probably must have received an email from my end. I am Deepsha Biswas, Community Manager at Pine Biotech. So you can just reach out to us. Dr. Amira, we do not give out certificate for a free session, but there are several free courses on our website, which I will be sharing the link to shortly, that you can take and you can use those certificates as well. And Noel, there are no certificates for this session, but as I again mentioned before, you can take some of the free courses which also come with certificates, and you can also use those certificates and put them in your LinkedIn profile, or you can put them to your CV as well. So we will be talking about that at the end of the session, and we'll be sharing those links as well. Yes, Abhimanyu, I will be sharing the link very soon. Yes, Iftikhar, we are going to be recording the sessions and they will be emailed to you shortly. So we are going to be beginning the session in a few more minutes. And as we see that everyone else is joining. Yes, Jeet. So we are also going to be talking about internship. So for today, right now, as we are going to be starting, we will be addressing the topic that we have decided to talk about today. Yes, Dr. Amira, we will be emailing you the presentation. If you have already signed up and registered for the event, then we have your email ID. If you have not, you can leave it in the chat box so that we can get to you. So let us start today's session. I will start by recording it on my computer. Thank you for joining again, and please keep your microphones muted and your videos turned off as we speak. And the chat box is again open for all questions. Welcome to this free webinar on bioinformatics for infectious diseases. In today's webinar, we will speak about bioinformatics for infectious disease research and present use cases for bioinformatics analysis methods that are used in viral evolutionary studies, antiviral drug discovery, as well as the emergence of drug resistance in bacterial pathogens. We will also showcase some of the latest developments on the Bioinfo platform designed for virology studies and introduce the upcoming Omics Logic training programs for those interested in effective utilization of bioinformatics for infectious diseases, immunology, and microbiology. My name is Beepsha Biswas, and I'm the Omics Logic Community Manager responsible for daily interaction with thousands of users that are part of our bioinformatics community managed by Pine Biotech. Over the past couple of years, we have seen participants from all over the world join our training programs, take online coursework, and utilize our big data analysis tools in various research projects. What we will discuss today and throughout this webinar is a result of hard work by an amazing team led by company co-founders Dr. Alfred Tauber and Dr. Leonid Brodsky. Dr. Tauber comes from a medical background and has extensive expertise in oncology, immunology, and biochemistry of inflammation, and has been recognized internationally for his generous support of many research projects and initiatives that support training and applications in medicine. Dr. Leonid Brodsky is the director of the Tauber Bioinformatics Research Center at Haifa and is an expert in bioinformatics and biostatistics. He has numerous publications in mathematics and bioinformatics, and over the course of his career has developed multiple novel algorithmic approaches to biomedical data analysis. The rest of the team you see here includes experts in bioinformatics, computer science, and training, and some of our team will be presenting today as well. In the upcoming Omics Logic program, you will have the opportunity to interact and collaborate with an international team of experts specialized in the application of data science to biological data sets. Let me introduce to the main speakers for today's session. Dr. Harpreet Kaur is a certified Omics Logic trainer and expert bioinformatician who will guide the hands-on sessions and assist participants with projects and analysis during the program. Her area of specialization is in genomics and machine learning. Dr. Mohit Mazumdar is a Romex Logic project mentor and an expert in big data computational analysis. Dr. Mazumdar has a PhD in computational biology and over 10 years of experience working with industry and academia. Ilya Brodsky is our co-founder and CEO at Pine Biotech. In his current role, he has extensive experience working together with industry and academic partners that are applying multi-omics integration and bioinformatics to a variety of biotechnology and clinical applications. I will pass it on to Ilya to talk about the increasing role of bioinformatics and data science in infectious disease research and introduce you to the types of challenges data analysis, data organization, integration, and visualization can solve for biomedical research projects. Over to you, Ilya.
Thank you very much, Bibsa. Share my screen. Thank you very much, Bibsa. My name is Ilya Brodsky. I'm the co-founder and CEO of Pine Biotech, and I want to share with you today a little bit of a perspective of what role bioinformatics is playing in different aspects of infectious disease research, and would like to also introduce a couple of tools that we have designed and developed for this kind of study to help understand the relationship between genomic and omic data in general with various aspects of infectious diseases. Genomics and bioinformatics have contributed immensely to our understanding of infectious diseases, from disease pathogenesis, or how the disease develops over time, to the understanding of how disease happens, or what are the mechanisms of interaction between the pathogen and the host, the spread of bacterial and viral antimicrobial resistance and antiviral resistance, as well as the host immune response. So how does our body respond to these challenges posed by these pathogens? As a result, infectious diseases has been one of the areas where genomic medicine has changed the way we interact with pathogens, detect them, characterize them, and develop treatments and prevention strategies for these important diseases. And so as a result, the application of bioinformatics to omics data has substantially advanced our ability to detect, study, and treat infectious diseases. And the achievements came as a result of a close collaboration, or an increasingly close collaboration, between computer scientists and biologists, placing bioinformatics, I believe, at the center of these current advances in this domain. And so I hope that in this presentation, you will also be able to see and appreciate the role of bioinformatics and how diverse bioinformatics applications in application to infectious diseases are. So there's an increasing need in mastering bioinformatics skills to be able to conduct such research. So the omics data that is now regularly being used to study viral genomes, DNA variation, host gene expression, protein abundance, and composition properties of microbial communities, they really require very different types of approaches to dealing with this kind of data to be able to effectively study and mine this data for patterns and really understand the pathogens and their interaction with the hosts. So today we'll take a look at several examples of these kinds of data sets and how they apply to key examples that we will discuss. So the role of bioinformatics in general, and in the context of infectious diseases, is really to allow researchers to access, process, analyze, and visualize these kinds of high throughput data. And so I will introduce several examples of how this was done successfully by researchers that are studying viral infections like the Ebola virus outbreak in West Africa, COVID-19 pandemic, as well as microbial pathogens that cause tuberculosis and malaria. We will briefly touch on viral genomics and evolution, as well as the analysis of gene expression patterns for detection of significant genomic variants, as well as important aspects of the microbiome and the virome. So really not just individual pathogens, but whole communities in a host that are being studied to find important associations with disease susceptibility as well as outcomes. In the last 20 years, availability of genomic data has led to an increasing understanding of how cells function at the molecular level and has allowed us to understand some of the major causes of deadly diseases that affect humanity around the world. With the current rise in viral outbreaks and their deadliness, there's a major focus on developing tools, data repositories, and data sets for better understanding of the emergence, prevention, and treatment of infectious diseases. So in fact, you can see here that in 2020, the coronavirus disease obviously has captured the attention of researchers around the world, placing it as one of the prominent causes of death. But nevertheless, many of the diseases that you see here listed have some kind of a pathogen behind them. For example, you can see here tuberculosis, HIV and AIDS, malaria, meningitis. So there are a lot of different hepatitis. There are a lot of different infectious diseases, diarrheal diseases that are listed here that are leading causes of death. Importantly, we can see how the field of virology and microbiology has developed over time. And you can easily see it in this illustration where we see in 2002, one of the first review of viral metagenomics, where the application of metagenomic sequences was highlighted to study viruses and the communities of viruses that they live in, and eventually leading us to 2020, where there has been an explosion in publications, all based on analysis of sequencing data. So what's happening, and this is an important trend that I wanted to highlight, is that sequencing technologies and the ability to analyze genomic data has led to an increasing abundance of structured data sets that are easy to navigate, to find data and mine that data, to study important and happening right now events, right? So if previously it took a long time before identification of a particular challenge to then finding the right data or developing that data set or a database, and then eventually coming with conclusions about the analysis of that data. Today, that's happening almost in real time. So regularly, researchers studying COVID-19 are generating data that is not necessarily as a result of an experiment, but just generating data from patients and from different types of experiments that are being conducted regularly, placing it into repositories that we all can access, and applying these tools to extract some meaningful patterns that immediately can lead to insights about how is this pandemic developing over time, and what are some of the important features or characteristics that we can focus on to prevent, treat, or understand this disease better. And importantly, I think that this trend over time has highlighted some of the important ways that we are starting to think differently about these pathogens, not only as individual elements, individual genomes, but really also as communities of microbes, of microorganisms, viruses, and bacteria that live together and interact and surround us every step of the way. And so there has to be a greater appreciation for the way that the host and the pathogen interact, leading to emergence of such deadly diseases as COVID-19, and making some parts of the population much more susceptible to be infected and lead to much more deadly outcomes. So really it has led all of this data and all of these data sets that have been collected have really changed the way that we think about viruses, bacteria, and other pathogens. And so one important emergence of this kind of an understanding has been the appreciation of the virome, which is the part of the microbiome and constitutes all of the organisms that inhabit us. So the microbiome includes uh, all the microorganisms that inhabit the human body, that include bacterial and archaeal organisms, as well as the microbiome and other members of the meiofauna, such as protists and metazoans. Studies of the virome are really in their infancy because it has only become possible recently to see the virome in large sequence data sets using bioinformatic tools that can detect relationships between viruses despite this high nucleotide sequence diversity. So in fact, the size of the mammalian virome is not even known. Although our own cells are outnumbered about tenfold by our bacterial microbiome, the number of viruses may be tenfold higher still. Human feces alone contain over 10 to the 8th or 10 to the 9th of viruses per gram. Additionally, the mammalian virome is continuously updated through rapid evolution of viruses and through exposure to viruses in the environment or in other mammals. So one of the leading reasons why we can now appreciate the role of individual pathogens, as well as study them as communities in their natural environment, is the ability to sequence and understand the variability of genomic data present in a given sample. So let's talk a little bit about one of the major technologies that has made this possible. And the technology, of course, is next generation sequencing. Now, the way next generation sequencing is used is also changing. It has been highly expensive and prohibitive in cases, but today the size of the instruments, the ease of use of those instruments, as well as the cost associated with producing high quality data have led to an explosion in next generation sequencing data. One of the challenges with next generation sequencing data is that it is still large and requires specifically designed tools to be able to extract meaningful patterns and find associations between short sequences and phenotypes. And so let's talk a little bit about what are some of these associations and what kind of metagenomic data are available to study infectious diseases. To study metagenomic data and to understand the composition of this data, there are really several different techniques that could be used at the sequencing stage. First, we can speak about targeted sequencing. In other words, we can select a particular organism or a type of an organism. We can prepare specific adapters that target sequences unique to that individual organism or group of organisms, amplify, and then study sequence variation, knowing what we are going after. The second approach is the amplicon. So amplicon sequencing is typically used to amplify a particular region, and that region might be useful to understand variability between different organisms present in a sample. For example, amplicon sequencing is hugely popular with metagenomics. When we speak about bacteria, the 16S ribosomal RNA or other genes have what's called hypervariable regions, or regions that allow us to study taxonomic composition of samples without having the full genomes sequenced and assembled, which is a major challenge because so many of the genomes do not have a reference. Another approach is to sequence the whole DNA or RNA. So we can extract all of the DNA or RNA present in the sample, sequence it, and then within all of that information to look for particular types of genomes or genomic elements and characterize what we have and quantify how much of it do we have. So these are very different approaches that allow us to extract very different types of information from a given sample. For example, one of the challenges with targeted sequencing is that we are not accurately characterizing how much of that particular element do we have in our sample. At the same time, with the whole DNA and RNA sequencing, it becomes very expensive because we are sequencing everything, and we are assuming that a lot of what we are sequencing is not going to be useful if we are focused on the study of a particular pathogen. So to understand how to find the right kind of data, or maybe how to sequence the right kind of data, and then how to analyze that data with the right kinds of methods, we really need to go behind some of the black box methods that are typically used in bioinformatics to understand the logic behind these analysis methods and apply that logic to a few different examples that will illustrate what this type of data can give us. For example, to study microbial and viral pathogens, several different types of sequencing have been applied with different types of analysis pathways or pipelines to understand different aspects of the pathogen or the microorganism that is causing a disease, to the community within which it is causing that disease. So what other microorganisms it interacts with, to then potentially understanding how that pathogen responds to different challenges applied to its natural environment. And those challenges could come from treatment, from the immune system, or from competition with other pathogens within its environment. So this kind of sequencing has been applied to both viral outbreaks to study them in real time and understand how they are changing over time, as well as to kind of persistent infections, including HIV, malaria, tuberculosis, and other pathogens that are regularly treated, and therefore they are highly likely to evolve and change the way that they interact with the environment because of those pressures. So here we have two examples. One speaks about viral outbreaks, where we can take a direct clinical specimen, and within that specimen that comes with all of the present organisms that are there, we could either amplify for specific viral sequences and therefore study the specific changes in the virus genome using Sanger sequencing, or using Illumina sequencing, we can take all of the present material, align the virus reads, or align all of the reads to a virus reference genome or a database of viral genomes, and then try to understand what kind of viral populations are present in a sample. In this case, we see a number of different types of applications of metagenomic sequencing to study different aspects of pathogens and how they interact with the human body. So for example, we can use it to identify a specific strain out of all possible strains to detect and really characterize what does that strain look like. We can also look for specific mutations or genes that are known to be associated with different phenotypes, including resistance to different treatments and drugs. And looking at the composition of different viral sequences present in a sample, we can even predict the emergence of a particular group of those viruses that will become dominant and will have an antiviral resistance characteristic to them. We can also look at the microbiome. So instead of looking at individual pathogens, look at full communities where we can compare healthy and sick, or healthy and different types of phenotypes, and really compare and contrast those conditions to understand how at the genomic level we can find different types of taxonomic organizational representatives, so different types of taxonomies that can help us understand how these communities might be related to a phenotype or to a condition. And then finally, we can look at transcriptomic data. So we can look at the immune response or the response of a cell to that pathogen to understand how that pathogen modulates the response, as well as to understand what kind of basic inflammatory processes and immune response pathways are activated and whether they are effective or what other types of maybe adverse response that might cause. So to really look at the variety of insights we can gain from such genomic data, we really need to understand what this data looks like. So here I have an example of what the sequencing produces. So the process of sequencing is really the process of generating short reads. Here, individual lines that you see are short reads that typically are mapped to some kind of a reference. And the mapping process is very dependent on the quality of these reads, of the sequences within each of the short reads that we have here, and the abundance of those reads. So the higher the abundance, the more confident we can be that we are finding very accurate signal. And then we can trust the changes that we observe in the signal to understand what kinds of mutations or variants are emerging in the sequence, and also what kind of variation we observe between different potential representatives of this kind of a sample. And
Ilya, you are muted.
okay, am I unmuted now?
Yes, Ilya, you are unmuted. Thank you.
Okay, so when we have this raw sequencing data, one of the major challenges is to really apply the understanding of what kind of variation we have to a detailed understanding of what this genome actually contains. For example, in the case of the novel coronavirus, we need to understand the properties of the different sequence elements or genomic elements that encode for different types of genes or protein producing genes, or even helper genes, so sequences that don't necessarily become proteins, but they assist in the process of replication. So from going to go from the raw sequence reads to be able to say something meaningful about what we observed and the variation that we observe, we really have to capture the data, have reference information about reference genomes and what those reference genomes code for. And then we have to piece together the different information that we have about the pathogen itself, how it interacts with its environment, and what is the response of the host that we can measure and quantify. So let's talk a little bit more about some of the analysis methods that are useful for this kind of genomic and transcriptomic data. So today we will speak about 4 different examples of methods of analysis that are useful to be able to study these kinds of viral and microbial infections, and let's think about what they can produce as we discover associations and patterns in such data. So we'll talk about analysis methods and tools for genomic and transcriptomic data analysis of host pathogen interaction. For example, methods to study relationships based on local and global multiple sequence alignment, what's called MSA. Phylogenetic analysis and evolutionary studies used for genome ancestry and population fitness. Chemical and structural implications of sequence variation mapped on PDB models, or models of proteins that we know of, and host pathogen interaction and treatment response based on transcriptomic data. To speak about these different tools, we have prepared different sections on the Bioinfo platform that you can easily explore and quickly apply to different types of data sets using the Bioinfo platform that has a visual interface. So let's go to that platform. Bipsa, if you could please post the link. on the chat. I'm going to open the chat. Okay, so please go with me to server.t-bio.info. Thank you, Bipsa. What you see here is a section of areas of analysis. Really, this represents different types of methods, bioinformatic methods, to understand and study such variation at the molecular level. So please let me know once you're on this link and it has opened up for you. You can just put number 1 in the chat or say, okay, or yes, I'm there. And I will know that at least a few of you are there. So we'll take a little look at some of these tools. Okay, excellent. Thank you very much. So I see that many of you are here. So now, just to explain, anyone that is interested in using some of these tools in the context of research or education, you're welcome to request an account. You can actually get a free account as a demo to understand how this tool works. But I'll briefly showcase today several aspects of this type of research that could be done on the platform using a visual interface. And then I'll transition a little bit to go behind the scenes and show you how these tools work. So first of all, what you see is a few different sections right here. And one important one is this access form. So if you are interested in using this tool in the future after the session, please fill out this information, and we will reach out back to you by email to understand what exactly are you looking for. For some of you, you might be our previous customers or participants of different programs. So if you do have your login information, you're welcome to log in. Okay, thank you, Bipsa. Okay, so let's take a look today at several different aspects, and I'll just first highlight those sections here so all of you can easily find them. So we talked about genomic variation, transcriptomic variation, analysis of phylogenetic relationships or phylogeny, and then also host pathogen interaction. So we have here several different sections. For example, here under phylogenetic tree, and you can open up here an example. In this example, we'll briefly speak about how you can go from raw sequencing reads to understand the phylogenetic relationship between sequences, and these sequences could be genes or whole genomes. We will also speak briefly about how you can conduct an analysis of viral host pathogen interaction. So here you can see an additional level of highly detailed molecular data that allows us to understand emerging mutations and characterize fitness of virus populations. We will also speak about viral quasi-species that allows us to look at point mutations, defective viral genomes, and also to link that to different types of genomic variation at the phylogenetic level. Finally, we will speak about transcriptomic data analysis. So using analysis of RNA-seq data, we will take a look at how you can understand response to treatment, response to pathogen infection, and characterize the immune response by looking at pathways and genes that are involved in that response. So first of all, let's look at what genomic data looks like and how we can find any kind of variation within this data. So if you don't have an account, you won't be able to do this at the same time as I do. But later on, if you are interested in repeating this demo on your own, we are happy to send you a demo account. So you'll have a login and a password to come in and do exactly the steps that I do. We're not going, for the sake of time, we're not going to do it together because we have a lot of participants. But in the future, after you review the recording, you'll be able to do that. So please request your demo account by filling out this form if you would like to do that. Okay, what I'm going to do is I'm going to go into this section for mutation variant. And I'm just going to give you an example how you can go from genomic sequencing, so sequences of DNA or RNA mapped onto the reference genome. So here we use bowtie2 to map to the reference genome, and then producing results in a visual format. So as a result of mapping on the reference genome, I'm going to get a SAM or a BAM file. And that SAM or a BAM file is going to show me what is in the reads. So let's take a look. Here I'm going to load the JBrowse application for the results of this pipeline. And the results will be loaded in such a way that I can easily navigate them and find what's in my data. Okay, so what's important here is to be able to understand what this data represents. So let's zoom in a little bit and we'll kind of take a look at what we can see here. As you can see, each sample represents millions and millions of reads. And these are individual reads that we can look into that have a sequence with A, T, C, and G. And those sequences are mapped onto a reference genome. So in this case, this is the human genome with variable coverage, allowing us to find variation or changes that are significant from the reference genome. So for example, here we can see this green little marker, and this marker represents a SNP or a variant that we can study. So we can look in and we can see what is this variant. So we can, for example, zoom in a little bit more here. Okay, and we can see what this variant is. Now, one of the challenges of analysis is to be able to map all of these reads to the reference genome, identify such variation, and then importantly, maybe most importantly, is to find what might be the association between this particular change and some kind of a phenotype. The other use case here, and let me do another quick demonstration here, is when our reference is one organism, for example, the host, but our input data we suspect has sequencing reads from both the host and the pathogen. So here I'm going to do some pre-processing on my data, and then I'm going to, again, map it onto, okay, sorry, not here. I'm just going to go quickly to another section. And I'm going to perform essentially the same process. I'm going to map on the reference genome and find the visualization tool and look at a different type of a sequence that was mapped to be able to identify a particular type of a genome. Okay, so here you can see again, this data is being mapped and I can see the reads, but importantly, when I go into the results, I will be able to find not only what is in my data, but also to find the individual reads and the percentage of how many of those reads were mapped onto the reference genome. Using such technique, I will be able to identify potential pathogens from a viral or a microbial database that I could then study in a more detailed way. Now, another section that we will look at today is a section that has to do with multiple sequence alignment. So let's take a look right here under phylogenetic tree. We can look right here. What is this section designed to do? What this section does is load in genomic sequences, and those genomic sequences contain maybe a variety of different viruses from the same family, or maybe it contains individual reads from patient samples. And we can then either work with FASTQ files based on sequencing reads, or we can align the genomic data to perform multiple sequence alignment and understand what is in our data, either as a relationship between multiple viral sequences, for example, or as haplotypes that you can study for variability and find association with a particular phenotype. So this is an example where we can look at haplotypes, or there's an example where you can actually look at, let me go back here, where you can look at the specific relationships between viral sequences. So using multiple sequence alignment and then converting those sequences to amino acids, and then understanding variability using all kinds of phylogenetic methods that I'll talk about in a second to really understand variability between genomic sequences. So let's take a look at the meaning of some of these basic analysis methods and how they could be used to study the genomic variation. So at the basis of all of these analysis methods is what's called multiple sequence alignment. And so here you can see the idea of multiple sequence alignment. When we take multiple reads or multiple genomes, we make sure that they align to each other in such a way that maximum similarity is on average achieved between the full sequences. We then identify specific variations, and those could be variations at the nucleotide level or at the amino acid level. And then we characterize those mutations to understand what types of mutations are we finding. Importantly, when we deal with next generation sequencing, we are going to find a challenge of characterizing those changes that only have low coverage. So the higher the coverage, it's important for us to achieve a high coverage to be able to be confident in the changes that we observe. Once we observe those, we can then start characterizing them by associating them with a particular phenotype. For example,
Hi, Ilya. We are not able to hear you. Probably a technical error on your end. Give us a minute, everyone. I guess there is a problem on his end. Yeah, thank you so much, Mohit. We'll just be back shortly. Okay, so I think Ilya has some kind of problem on his end, which I will be having a look at. So the session is not over yet. Yes, hi, Ilya.
yeah, sorry about that. Something happened here with my network. Let me try sharing again.
Yeah, thank you.
Can you see my screen?
Yes, Ilya, we can see your screen. Thank you.
Okay. Okay, sorry if my network here for some reason became a little bit unstable. So again, to be able to understand what process is causing these variants, or what are these variants associated with, we need to touch on the biology of the pathogen itself, because in each case, with each different pathogen that we're talking about, there's going to be a variety of factors that we need to consider. For example, when we speak about viral infections, where we have a basic entry of the pathogen into a cell with the objective to hijack the host machinery to produce viral proteins, assemble them, and eventually produce multiple replicates of the same virus. Now, to really go into the detail of this process, not only is the entry different between different types of viruses, but also these viruses are not homogeneous. They actually represent a community of various types of sequences. And so one of the questions is whether one of those particular types of genomes that are entering the cell eventually lead to a variety of the different types of pathogens that exit the cell, and what are the different immune responses or strategies that the virus has to adapt and avoid or evade the immune response of the host. So when we study genomic variation for the different pathogens, we are actually thinking of how these particular mutations that we observe in the sequences that are captured represent a relationship between the ancestor or the sequence that entered into the host and the present day species that we observe. And the way to understand the relationships is to map the different groups or genomes that we have into what's called a phylogenetic tree. This phylogenetic tree can help us map the differences between genomic sequences in such a way that we can associate it with a process of going from a single ancestor to then dividing up into branches, just like a family tree would, that we can study for variability and distance, evolutionary distance between each one of our members in the data and the potential common ancestor that they might have. So what kind of information can such an analysis yield? This is an example of a phylogenetic analysis of the 2016 outbreak of the Ebola virus in West Africa. Here you can see, and this has been done obviously for many other viruses, including COVID-19. So here you can see from 2014 to 2015, we can see one single type of the original pathogen that started this epidemic, and eventually split up into multiple clades. And some of those clades were more fit, they were more capable. Therefore, we have observed them to be more present in later days. And some of them we can now start thinking about, well, which geographic locations were they more prevalent in? Obviously, in each one of our geographic locations, we have different types of phenotypes for people, for treatments, for the way people interact, for the different death rates, for the infection rates, et cetera, et cetera. So now we have an ability to structure our data and study evolution of the virus, its ability to adapt to different types of responses, the common origin, right? One of the questions could be, did it all start from just one single location? And which location did it start from? And which location did the transfer happen at? As well as the divergence, right? So what might be some factors that contributed to this highly divergent phylogenetic tree at the end of that process? Now, as I mentioned, this type of analysis is also very effective, not just for this particular case of the Ebola virus, but also here beneath, we can see a similar analysis for COVID-19. So for COVID-19, an initial analysis was largely focused on how we can study such type of variation between different known species of the coronaviruses like SARS-1 and MERS, as well as some of the novel emergent pathogens that we are starting to see beyond that first characterization of COVID-19. For example, in other animals, what might be a potential host reservoir that we should be cautious with. So one of the things that we can do with such information is not only look at sequence similarity, but start looking at which particular regions of the genome show us more or less variation, and what kind of consequence does that variation have at the amino acid level, at the level where it actually codes for a protein, therefore having a functional capacity. So one of the things that have emerged from the very beginning is that such variability in the case of COVID-19 was not uniform. It was not the same across the full genome. So if you look down here at this figure C, you can see the nucleotide identity, or how similar different genomes are to each other or to a single reference. And you can see the dips, right? So these are lower areas that show that there's much less consensus. So they're more divergent, they're different. And the differences now have to be mapped to the different particular proteins that are encoded in those genes. So probably many of you have already heard about the spike glycoprotein, the protein that is responsible for viral entry into the host cell. And we can see that this particular dip happens right here with the spike protein. And here you can see another dip that is less than that, but also this happens at the N protein. So now we can think about, well, what might be the consequence of such changes in association with protein function? When we want to understand protein function, there are several ways that we can deal with that variation, ultimately relying on, again, genomic data. So we talked briefly about how nucleotide level similarity can inform us about amino acid level sequence similarity. When we look at the sequence similarity, we can look at genomic context or features, right? So if we know where this particular genomic element is being expressed, or what is its functional class, or we can compare global and local sequence similarity. These can be tools in our toolkit to study amino acid variation based on nucleotide variation found directly in the genomic data. The second option that we have is to understand the 3D structure. So ultimately, when we have a sequence, that sequence gets translated into an amino acid sequence because it folds. And the amino acid folding happens based on physicochemical properties. So how the different amino acids could be put together into an actual structure that is going to become functional. Now, here we also have some tools to identify different types of functional elements or motifs that we know and have characterized in the past. We can also look at the actual structure of this protein, if it is available, and try to understand the implication of nucleotide variation on the properties of the overall structure. Another method, and we'll talk about this in just a second, is to generate high throughput data. So we can generate data on gene expression, for example, by taking out a particular gene or a protein or a portion of a protein, or introducing a mutation that will cause a stop codon, that will lead to a stop codon and shorten the protein. And now we can study differential gene expression analysis, which will reveal how the expressed properties of that protein have changed after we have introduced that modification. So really, those are the three elements that we can connect and combine together to have a full overview of what is going on. Now, importantly, a lot of this data is already available online. So we talked a little bit about different tools. Let's speak a little bit about different resources that you can find to understand what kind of genomic information is available and how you can start right away to develop an idea or a project for a particular type of a pathogen. Okay, so let's go quickly to NCBI virus. please go with me to NCBI virus. Just going to paste the link right here in chat. Okay, please let me know once you've arrived at NCBI virus. Okay, thank you, Michael. If you could please put in chat that you've arrived here. Yes, please put number 1 or number 2. Excellent. So now that you're here, right, the reason I'm saying 2 is because we already had our first link, which was the server where we analyze data. And this is now our second destination. The second destination is, where do we actually find the data? Okay, so for those of you that got here, obviously you have a quick access to SARS-CoV-2 data. But importantly, the data set includes a variety of different pathogens, and here we're just focusing on different viruses. So here we'll quickly see a number of different viral sequences that are available. So if you look on search by virus, if you click on search by virus, think of any virus: coronavirus, influenza, hepatitis, Ebola virus. So really any virus that you can think of, and just place it right here. I'm just going to place here Ebola virus. Just give it a couple of seconds. And you will see that this drop down appears where you have different subtypes of that family, the Ebola virus family, and here are the different types of the Ebola viruses. So for example, we can see here Zaire Ebola virus. So once you click on that particular type, you can now see the different data sets that are available. So by looking at this data, please let me know how many nucleotide sequences are available for this Ebola virus, for the Zaire Ebola virus. You should be able to find on this page how many nucleotide sequences are available. Excellent. Thank you. Chandra Prakash was the first one, so you get a special call out there. Okay, so 2,938, so almost 3,000 viral sequences for Ebola virus, for those of you that are interested. Let's take a look, and instead of the Zaire Ebola virus, now we can just remove this, and here add another plus, and let's search for coronavirus. So you can see the Coronaviridae family, and now tell me how many are available for the Coronaviridae family. All right, great. Noun Poon got it first. So special call out to Noun Poon, 69,000 sequences. Now, who remembers, when did the Ebola virus epidemic really start, as a big concern? How many of you remember the Ebola virus outbreak? Okay, 2008, 2016. Okay, really 2014 is when it started. There have been outbreaks before that. Okay, so I see Mohit Mishra got the first right answer there. 2014 to 2016 was really the outbreak, so it started around 2014. Now, if you think about it, we're in 2020, that's just 6 years ago. So we've had this ongoing research of the Ebola virus, and the sequencing change, really the way that sequencing was used directly during the outbreak is that first outbreak of Ebola virus, because of several different reasons. So in previous outbreaks of the Ebola virus, like in the 1960s, mentioned here by Abir, 1970s, I think is when it was, you had very expensive and highly laborious process of generating those sequences. And so as a result, those sequences accumulated fairly slowly. But in 2014 to 2016, there was also an emergence of Oxford nanopore sequencing, which was very affordable. It was not very accurate, but you could literally take it to the field and take a sample and sequence it right there in the matter of maybe half an hour. So as a result, there has been an emergence of this real-time sequencing data about a pandemic that was going on. And many, many labs around the world were focused on analyzing those sequences to extract meaningful information that will guide the detection, characterization, treatment, and prevention strategies, ultimately resulting in a vaccine and not so long ago, an antiviral treatment for the Ebola virus. As opposed to today, right, you can see the rate at which this is happening, right? So remember, 2014, 6 years ago. Today, we're in 2020. So from 2020, from maybe December 2019, but ultimately in 2020, so we're just talking about a matter of months, we already have almost 70,000 sequences. So when we have 70,000 sequences, the challenge is not in generating the data anymore. The challenge is in analyzing this data to understand what it means. And so that's why I think for any one of us that is interested in such an understanding, not only do we have really an explosion of data that is available on viral genomic sequences, on whole bioprojects that give us access to different levels of information about both the pathogen and the host, as well as the specific characterization of even protein sequences that we can access through PDB. So we're not just looking at the level of genomic similarity, we're also starting to look at what does this genomic information tell us about the variation of key proteins of these pathogens that actually interact with the host proteins as well. So here you can see the spike protein, and the spike protein mapped with mutation frequency based on the analysis of different types of coronaviruses, and specifically COVID-19 compared to low mortality rate coronaviruses, where we can start to get an understanding of how different elements of this spike protein, the ones that interact with the binding, the ones that do cell fusion. So we really can apply our understanding of the real world situation with that genomic data, right? So combining those two types of data together. And ultimately, this kind of understanding can have a profound effect on our ability to characterize transmission, detect when the transmission happened and how it happened, how we can think about different types of intermediate hosts and potential intermediate hosts, as well as thinking about the actual biology at the molecular level, where we have the receptors, the immune response and the interaction with the immune response, tissue tropism, so how the virus adapts to different types of tissues and tissue barriers to ultimately design different types of intervention strategies to try and address the current epidemic. Now, importantly, that we need to link all of this information to a quantifiable process. And in this case, we can have a quantifiable process of the rate of mutation linked to our biological understanding of how the rate of mutation and the mutations themselves affect the particular regions on the proteins that we can study. For example, we just looked at the spike protein. One of the regions within the spike proteins that has the most variability between the high and the low death rate associated coronaviruses is what's called the receptor binding domain. This receptor binding domain is really a part of the S1 subunit that is located within the spike glycoprotein sequence. So it has several different functional domains. And these functional domains have different rates of mutation. So those variations that we observe at the genomic level, they are happening at different rates in different portions of this protein. And to understand what they might mean, we can really start taking a look at the receptor. So this is the host receptor right here that you can see in the blue is the ACE2 receptor. And you can have the spike protein actually bind it. Now, this ACE2 receptor actually sits within the host cell membrane, just like that. And this sodium-dependent neutral amino acid transporter is what holds this ACE2 receptor in place. When the virus comes, this is the spike glycoprotein. It is protruded from out of the membrane of the virus, and it has this particular receptor binding domain, which is colored in red here. Amazingly, we have all of these structures already available. And so because we have all of these structural elements available, all of the genomic data that we have can be highly informative as we study protein-protein interaction. So the interaction between the spike protein and its receptor binding domain to this particular protein called ACE2. Okay, so I'm going to pause here for a second, and I wanted to see if there are any questions that I can address at this time.
Ilya, there are a few questions which I have. So we have one from Kehinde Adeshina. Please, what's the best sequencing platform for microbiome analysis? And do you have any guide on using nanopore sequencing platform for 16S rRNA sequencing? I would be working on this for my MSc research.
Okay, thank you for that question. Yeah, so it's difficult to say what is the best sequencing platform because they have different aspects of quality of the actual sequence, the cost, which is an important factor, so you determines how many sequences we can make, and also how easy it is to work with that data. So basically there, I would say, are 3 or 4 different types of sequencing data that you will find for this kind of application. There's Sanger sequencing, which is the gold standard for long sequencing. You can have very accurate sequences, but the problem is that you need a lot of annotation to be able to produce those. There is Illumina sequencing, which allows us to find really a balance between high level of accuracy and short reads, but it is a little bit expensive. However, there is MySeq, Illumina MySeq platform that allows us to, and it's typically used very popular for metagenomic data analysis, so 16S ribosomal RNA, where you have a long read, about 300 to 350 base pairs long, and you can use those to capture 1 or 2 hypervariable regions. So for example, V4, V6, V4, V9, you can use different combinations to study taxonomic variability. Then you have Oxford Nanopore, and I would also put PacBio maybe in that category. So Oxford Nanopore is very affordable, easy to use. The data has it's not such high quality, so you do have a lot of variability, but it is useful to characterize populations, right? Because in the case of metagenomic studies, you're not looking at individual sequences, individual genomes, you're really looking at taxonomic variability and composition. So I think that is a very good platform to use for metagenomic sequences. Okay.
Yeah, there are a few more questions.
Yes. Okay.
So Noel has a question. I'm planning to do a whole genome sequencing of Campylobacter. Do you have a user-friendly platform that may help me in analysis of obtained data?
Yeah, so what I showed today, and I will give another little example with the Ebola virus here, there are multiple different ways that you can use genomic sequences to understand what you have found. For example, you might be focused on, as I mentioned, phylogenetic analysis, so divergence of present day samples from a potential common ancestor. You might also want to look at differences between different types of samples that you have, maybe associated with a different phenotype, like their geolocation or resistance to different types of treatments. So importantly, you have to have the data in certain formats to be able to use these different types of analysis. One part of that data is the genomic sequencing. The other part of the data is what do you want to associate it with, right? So I'll give here in a couple of minutes another two examples on Ebola virus and on tuberculosis to kind of illustrate what is needed for those analyses. But yes, once you have the genomic sequences, that is a big part of the story, and you can do a lot with that sequencing already.
Okay. Okay, Ilya, so we do have a few more questions. One that we had from Dr. Manav Jain, the certificates that we offer for the program, he has a question regarding that. If they have any traceability like barcode or anything else.
Okay. Yeah, so we do have a permanent link for those certificates. I will pass it on to Bipsa in just a few minutes to speak about the training program that will start in November for bioinformatics and infectious diseases, where many of these methods are going to be covered in greater detail with access to some asynchronous material as well as projects that we have curated to be able to illustrate some of these methods of analysis. But right now, I just wanted to make sure that I address the questions about the topics that I was talking about. I see here in the chat, okay, so regarding, okay, I see here, I read a few papers regarding neuropilin-1 along with ACE2 receptor that helps in COVID-19 infection. Can you give us information about that? So again, there are specific use cases. We will go into much greater detail about the coronavirus, the Ebola virus, tuberculosis, malaria, some other pathogens in our upcoming program, where we will take the available data and actually try to mine that for patterns. I think that the key here is that in the context of bioinformatics, we will not just analyze published research, so how other people have done something, but we'll actually take that data and analyze it ourselves to make sure that, first of all, we can arrive at similar conclusions that they have, to understand the logic of how they arrived from raw data to such conclusions, and then ask additional questions that maybe they have not previously asked about how we can take that research further. So I hope that for those of you that are joining the session, it's really to give you a glimpse into what kinds of data sets, what kinds of methods of analysis, what kinds of questions we're going to cover and highlight. And so maybe before we jump into the program, I'll just briefly complete my slides and I'll pass it on to Bipsa to speak a little bit more about the registration process and the different resources. Okay, so I briefly wanted to mention that when we have genomic data, right, and I already illustrated the process of visualizing that data, that just the visualization itself is not very kind of clear, because we need also statistical analysis of that data to be able to determine which ones of the different mutations or variants that we find are actually functionally significant. And that was a major objective in the process of the pandemic or the epidemic of the 2014-2016 outbreak, to determine mutations that might be associated with airborne infection, which in case of Ebola that has a very high mortality rate was a great concern, as well as what types of variants are going to have an impact on the potentially developed vaccine. So as we will cover in the program, we'll talk about the process of taking those same mutations that you can see that we visualized, and then doing some statistical analysis for variant calling and characterization of the genomic variants to be able to identify the mutations that are most significant. As you can see right here, this is the actual Ebola sequencing data that were later on used in the proteins that were included in the testing of the vaccine that was designed for the Ebola virus. Now, the same type of an approach, I mentioned that data on one side could be genomic data, right? And we just kind of use here reference proteins that we have structures of to understand the impact of those variants. We can also look at a different approach. This is an example of tuberculosis. Again, we have here visualization and then characterization of mutations. But on the other side, if we have data about response or data about emergence of different types of mutations associated with antimicrobial resistance, we can start thinking about linking those two data using two different approaches. One approach is what's called GWAS, so genome-wide association studies, associations of multiple positions on the genome with a phenotype, which require a lot of genomes, right? So if we have hundreds or maybe even thousands of genomes, let's say from tuberculosis, and we have incidence of which groups of genomes are associated with different types of treatments and different types of emergence of single drug or multi-drug resistant strains, we can find statistically significant associations with regions. So you can see here either single mutations or whole regions or multiple mutations together in different locations that cause such resistance. Now, obviously, these are just a few examples, and we don't have time in the context of this one webinar to go through many examples in great detail. But in the program and in all that we do, what we try to do is take data from current and peer-reviewed publications, analyze the methods that the authors have used to come to the conclusions that they have published, and then to try and extract additional data sets and additional methods to develop independent projects with our research fellows. And so I do want to mention that the program that we have been discussing today is really also kind of enriched and expanded by the research fellows that have joined our research fellowship, and I'll let Beepsa talk about that in a second, that have been able to find very useful case studies and really look at both artifacts and signal in the data that they have submitted on NCBI and other publicly available repositories to really develop interesting findings from the different projects that you can find online. Now here you can see some of those examples. For example, this is a student submitted project proposal that we go through together during this upcoming program. And I'll let Beepsa maybe take over here in a second to speak about some of the program resources that we have prepared for this upcoming program called Omics Logic Bioinformatics for Infectious Diseases. So really what I showed today is just a glimpse of a Omics Logic full program that will focus on some of these different examples of coronaviruses, Ebola viruses, rhinoviruses, tuberculosis, and malaria-causing pathogens to develop an understanding of how to logically connect the tools and the data to produce interesting results that are going to be of significant value to the virology community. So to speak about some of the program resources, I am going to pass it on to Beepsa to explain how to register for the program and how to gain access to all of those resources. Over to you, Beepsa.
Thank you so much, Ilya. Let me share my screen here again. Okay. So before we start talking about the different resources, I just want to give you an overview of the upcoming program, the Bioinformatics for Infectious Diseases. So over the course of the past several years, our team has been collaborating with a number of academic institutions around the world that have participated in content evaluation, curriculum review, and project design. As a result, the Omics Logic programs have been thoroughly evaluated and are known to lead to excellent outcomes. Importantly, the programs are designed to support students with minimal bioinformatics background, including biology and biomedical studies majors, as well as faculty interested in bioinformatics, data science, and biomedical research. We have designed this program to introduce the data, resources, and tools that you can use to apply bioinformatics to publicly available data on infectious diseases. In this program, we will understand viral diversity and its role in epidemic infectious diseases that keep re-emerging, including zoonotic transmission, transmission between humans, and the process of viral and bacterial disease development, as Ilya demonstrated earlier. We'll learn about bioinformatics analysis approaches to study viral genomes and understand the relationships between viral strains, haplotypes, finding differences in sequence data, and seeing the implications for drug and vaccine design. You will gain hands-on experience by analyzing curated datasets from public domain collections, guided by experts with bioinformatics experience and knowledge about virology. The goal of the Omics Logic program is to provide training in bioinformatics, enabling independent research guided by mentors and peer example, developing a growing community with shared passion for data-driven research and appreciation of citizen science, enabling students, clinicians, and faculty of all backgrounds to develop novel and independent research using latest technology in life and data sciences. So we will combine online sessions like we are having one today, then self-guided study materials, which I will take every one of you over in just a few more minutes, for an immersive experience that has proven to be effective in our well-known and respected Omics Logic programs. Together with our collaborators, we have been building a growing online community of bioinformaticians in training, ranging from high school students to faculty and researchers in top universities around the world. And in all of our programs, this community of bioinformaticians, students of biology, have access to these tools which are here. So Ilya has already demonstrated the server T-Bioinfo platform, and I will also demonstrate a few of these other platforms and show you the resources that you will have access to if you are joining this Omics Logic program or any of our other Omics Logic programs. So the Bioinformatics for Infectious Diseases is a 2-month program that will include a review of how to use analysis tools, provide examples of infectious disease-related studies, and then offer support to those interested in working with public domain dataset of their choice. We will meet twice a week for discussions, Q&A sessions, and hands-on demonstrations that everyone can participate in. So let us review the schedule and the registration process together. So I am going to be going to the registration link. Here I am pasting it on the chat so that you can also go along with me to the registration page. So this is the Bioinformatics for Infectious Disease registration page. And this program is starting on the 9th of November. It's a 1-month program and will go on till 2nd December. Every week we'll have 2 sessions and they will be timed at 9 a.m. CST. So 9 a.m. CST is in Indian time, 7:30 p.m. IST. And you can also convert it to your local time if you are not from any of these regions, which is either the US or in India. So let us just move down. We have a program overview of what the program is about. And here you can see the entire syllabus for the different sessions. So you have the different session topics mentioned here and the different dates of the sessions. So usually these are all 1-hour sessions like we are meeting today, where our mentors will give you an overview of these different topics and go in depth on the analysis. And you will carry out the hands-on analysis on your end as well, because you will have access to the resources which I'm going to be showing and which also Ilya demonstrated earlier. And we will have different sessions as you see right here on these topics. And you see that there are necessary gaps between the 2 sessions so that you can go through the asynchronous course materials that are offered as part of this training program to help you learn on your own and come up with any questions that you have. And So the resources that you will have access to, I will be coming to that one more time. So how do you register for the program? You can click on Get Started. And there are 3 membership options which you can choose from. So the different membership options come with different benefits. So the beginner options will give you access, as you see, to all the different courses that are going to be mentioned in the program. It will give you access to the code playground, which I'm going to be showing in just a few more minutes, and also the interactive sessions and the Bioinfo platform. The intermediate membership option will give you access to a one-on-one mentor session where you can request a mentor to help you out if you have any questions, and have a dedicated one-on-one meeting with that expert or the bioinformatics mentor. While the advanced membership gives you access to another month, like not just till 2nd December, but till 2nd January, you will have access to the platform and the resources so that you can work on your own project. So I know that I saw in the chats that many of you are interested in working on a COVID-19 project or a similar project on infectious diseases. So this advanced membership would be for you, where you get another month of access. Now, for students and scholars and anybody else who would want to join the program and may think that you cannot afford, we have a scholarship program as well, and I'll be sharing the link shortly in the chat. So before that, let me move on to the different resources that we are going to be discussing here. So the first is the server Bioinfo platform. We all saw how the server Bioinfo platform works. So you will be able to gain access to the platform, which will not be a demo access, but be an educational access where you will be able to use data from public domain data sets like Ilya was talking about earlier. And we will be carrying out hands-on analysis. So all that Ilya has demonstrated in today's session, the mentors would be taking you step by step on the different logic of analysis in these sessions. The second platform would be the Code Omics Logic platform. So you can still start by registering yourself for free on this platform. There are a few courses available, and this is our platform for practicing R and Python in your browser. So you do not need to install an R and Python, but you definitely can do that. So you have courses getting started with bioinformatics, and I know that a lot of students who are just learning or starting to learn about bioinformatics are also very much interested in data science. So we have developed this platform where you can easily learn about data science in the respect of biological data. So you will also have access to this platform. And you will have access to what we call an organization page. So here I have just opened a tab on the courses. So these are the different courses that you will have access to besides the live training sessions. So you will not only have access to the program live sessions, but also the courses for your own one-on-one learning and self-study. And each of these courses have an individual certificate that you can get after you complete the course. Yes, thank you so much, Ilya. And I also wanted to talk about the fellowship program just for the people who are interested in an online internship. So this is the fellowship link. And in our research fellowship program, we have students joining us or participants joining us for 3 months or 6 months research fellowship. And the focus is mainly on working on their own projects related to areas like neuroscience, infectious diseases, cancer biology, and more. So if you have any inquiry regarding the same, you can write to us at marketing@pine.bio. You will see all of our Omics Logic program comes with, again, basic or advanced. And the difference is again on the different level of access that you get to the Bioinfo platform. Here, the advanced license will allow you to use your own data, not just public domain data, use your own data to work on your own project. And you can either do it for 3 months or you can choose to do it for 6 months. That is completely up to you if you are interested. And here is the scholarship link that I was talking about, that students, if they are interested and they hope that they want to join the program, you can fill it up. And you will see that at the bottom of the page, you will have to provide these details, and you have to also mention that why you're applying for the scholarship, and you have to mention a reference from your college or university. And we will reach out to you after you fill up this form. And you can also choose the different programs that you are interested in and write down about it. So this is something that you can apply for. And as Ilya was mentioning and he shared earlier, we have 2 groups, which is the LinkedIn group where you can join. And we have the Facebook group that also you can join again, because we keep posting updates there. And as we shared the link to our YouTube channel earlier, so we also have our YouTube channel where you can actually see all the different videos. So we post a lot of informative videos from not just our webinar sessions, but also on different topics that you can explore right here on the platform. You can see a review. So this is a review from one of our project interns who worked with us on a project in glioblastoma. So you can take a look at that as well. And as I have been showing earlier, this link which I shared is a list of project examples that people have been able to do once they joined one or either of these Omics Logic programs. So if you scroll down to the page, you will find that we have a lot of posters that were submitted and presented at different webinars, at different conferences. You can see the different projects here by one of our interns who collaborated with 2 of our other participants from Sri Lanka, and they came up with a project on SARS-CoV-2. So definitely, if you're interested, you can do a project on SARS-CoV-2 as well. Then you'll have more projects, the glioblastoma project that I just showed you earlier. Then you have more projects that you can explore. So please feel free to explore the different projects that are available and that have been actually independently developed by participants, where we from Pine Biotech and our mentors helped them throughout this process. And if you are just coming to the platform for the first time and you just want to explore the different resources, you can also take a look at our different membership options as well. So we have a very basic membership option, which is just for $10 USD per month. It will give you access to the Bioinfo platform. You won't be able to upload your own data, but you will be able to access all the demo pipeline and run from them. And also you will be able to access 12 courses on our platform from different sections like transcriptomics, genomics, metagenomics, and epigenetics. So you can consider this option as well. We also have a specialization subscription, which will give you access to all the courses, all the available courses on our platform, and not just selective courses. And we also have an unlimited $99 research license. So the unlimited membership will give you access to a 1-month research license to the platform, where you can actually upload your own data that you have generated from a lab, and you can analyze that data and also have our team help you in that process. So you have these options as well. But again, if you want to join the Bioinformatics for Infectious Disease program, then here is the link, and we will also be emailing you the link to this program. And if you have any questions related to either of these resources that we discussed today, or any of the other things that you may want to seek our help about, then you can please email us marketing@pine.bio, and we will reach out to you. So now we will be taking in questions. So do we have any questions?
Yeah, thank you very much for the presentation. I just wanted to mention that we also have a feedback form for those of you that are joining and would like to provide us with any kind of feedback. I'm pasting the link to the form here in the chat. Please do join us for our upcoming free webinars, so you can stay on these social media networks to that Beep's already mentioned on LinkedIn and Facebook to learn about upcoming free webinars. For those of you that are on the Western time zone, US time zone, we will have another webinar for infectious disease research on Friday in the afternoon. So you're welcome to join that one as well. And we regularly have these free webinars and workshops online that many of you that I see the questions about multiple sequence alignment or different types of data, we'll be addressing those there as well. So again, thank you very much for joining today. It was exciting to see that so many good questions came and so many different types of analysis ideas and projects are already emerging. We hope to see all of you in the upcoming program to be able to go into these different types in detail, and looking forward to interacting with you so that we can learn more about your interests, which I hope you will be able to share with us through this form. Thank you again, and thank you, Beepsa, for doing this session, and hope to see all of you in the near future. Yes, there are courses about genomic analysis and using machine learning. We have that on the EDU portal, as Beepsa showed. There's a whole section on genomics where you can learn about genomic data. And there's also a whole series of courses on machine learning as well. There's an introduction to machine learning on the EDU portal, and there's a whole series of courses in R and Python where you can actually learn about the models themselves, and you'll be able to practice right there in the code. We are just going through our program on data science as well, so we'll have that repeated in the future. Again, thank you, everyone, and I hope to see you again soon.
Thank you, Ilya, and thank you, everyone. We'll see you soon. Thank you for joining us today. For any questions further, please email us marketing@pathogen.com.